Hello and welcome to another module in the MIDD training curriculum. This is the second part of a series on model-informed decision-making in drug development. My name is Mark Gasronguay. I'm the founder and CEO of Metrum Research Group. In our session today, we'll be applying MIDD strategies to decision-making. I'd like to acknowledge contributors to the content in the presentation today, including our Metrum Research Group team of scientists, the MIDD Training Steering Committee, and other collaborators in industry, government, and academia who have influenced my thinking and have contributed to some of the concepts and principles we'll be sharing today. A little bit about me. I'm trained as a professional pharmacist with a PhD in pharmacology and a postdoctoral fellowship in pharmacometrics. I've been applying modeling and simulation to problems in drug development and clinical therapeutics for the last 30 years or so. That spans a career that covers academic, industry, contract services, nonprofit, and government sectors. I'm passionate about the work that we do. I believe that this is a great way for biomedical innovators to make decisions and gain insights. I'm a strong proponent of open science, and I'm most proud to be part of the team at Metrum Research Group. Really fortunate to work with that excellent group of individuals for the last 18 years and with all of our collaborators along the way. In addition, I've been privileged to work as an adjunct and graduate faculty member, learning from and mentoring multiple students and fellows over the years. There's a bit more detail here, which you can review on the slides on your own. And let's get started. So continuing on from the prior module, we'll pick up where we left off in reviewing the MIDD strategy. Then we'll dive into a bit more detail, thinking about the questions, quantitative criteria that we'll use to assess our model-informed approaches, consider the relevant knowledge sources available to apply an MIDD approach, opportunities to fill gaps in the knowledge or make assumptions where necessary. We'll look a lot at how to use simulation, which is the theory applied in action from a mathematical model, to evaluate scenarios and explore different decision paths. We'll wrap up with some assumption checking and some additional examples of MIDD in practice. By way of review, the MIDD strategy that we outlined last time is one that starts with questions. We start with questions both qualitatively and quantitatively because the details provided in a quantitative specification are really necessary for us to understand the type of information that we'll need to support that, the specifics about the models, and the simulations that we'll take on. Next, we'll proceed to identifying knowledge sources and filling gaps in that knowledge. There are a couple of ways to fill gaps. We'll see that we can do so by making assumptions where there is not much of an option, or potentially integrating data sources and interpolating using model-based methods. From there, we apply those derived models to simulate and address the particular questions or scenarios of interest, evaluating the results of those simulations in the context of decision criteria, then checking assumption and sensitivity to any of those assumptions and uncertainties, finally arriving at some decision or guidance that is model-based. Of course, this is an iterative process. As additional information, additional data become available, we can run through this cycle again or apply the cycle to a new set of questions. Let's review some of the key characteristics of a strong MIDD strategy. MIDD strategies organize around a series of questions. Those questions are supported while also aiming to advance the theoretical understanding of the problem. We quantitatively integrate knowledge and bridge knowledge gaps, allowing for the exploration of alternative options or scenarios. A strong MIDD strategy is transparent about underlying assumptions, assesses sensitivity of conclusions to assumptions and uncertainties in the simulation model, and delivers insights with the associated probabilities of desired outcomes. We'll highlight each one of these attributes as we see them in examples throughout this session. So let's start by defining the questions. When making decisions using model-based methods or any other method, defining the question is the key first step. In a model-based approach, questions guide model development and evaluation needs. They identify which sort of model structures and components will be necessary, which features of the data are important to reproduce accurately, and they also guide the simulation model design. What sort of structure or components are needed? Will there be an extrapolation beyond the observed range of data, or is it interpolation between similar data features? Some of the questions that a development team might be faced with are simple. Things like, what dose is necessary for efficacy? Is toxicity a concern at this dose? What fraction of the treatment population will be adequately served? Will this trial succeed and meet expectations? These are typical questions in a drug development program, and also the key part to beginning some sort of decision support. When working with development teams, those of us who are applying model-informed drug development principles will want to elicit and understand the key details underlying these questions. It's imperative that those who will work to implement the MIDD strategy understand the question accurately. As Dan Rome puts it here, whoever best describes the problem is the one most likely to solve it. That goes further than just hearing the initial question that's been communicated by a development team. It's actually investigating that question, probing it a bit further, understanding what are the really important components. Those might be obvious to the decision maker, and sometimes they're not, at least not initially. But it's this sort of interaction that will help to elicit the important details and components of the general question that's just been asked. Typically, this sort of definition of a quantitative translation of the question requires specific definitions of clinically relevant effect size, response rates, time points, endpoints, and so on. So rather than asking at which dose will this drug be effective, the question might be which dose leads to an average response rate of 85%. Or when considering potential toxicity, you might consider a dose that reaches no more than 10 millisecond increase in a particular endpoint. Or maybe even thinking about a fraction of the population. At least 90% of patients are required to achieve a particular target concentration. You'll see more of these examples as we continue. Here's an example of a key question in a quantitative translation. The question is a simple one. Is toxicity a concern at this dose? But with further inspection and evaluation of what's important, that question, that simple question, could be translated to something more specific. For example, to be competitive with the standard of care, the toxicity incidence must be less than 30%. What's the probability that we're in that ballpark? Furthermore, to really exceed expectations, we'd love to see a tox incidence less than 12%. What's the probability of achieving that goal? On the right-hand side, we see a probability distribution of the expected toxicity rate at a particular dose. This is a model-generated probability distribution, which is centered somewhere with a mode around maybe 3 or 4%, but with a skewed distribution that allows some of the higher incidence rates to be plausible. When we compare this distribution to the particular quantitative criteria that were defined, 30% rate and 12%, we can then make probabilistic statements about the outcome at a particular dose. So for example, what's the probability that the incidence rate is less than 30? That's quite large when you see that about 99% of that distribution lies to the left of 30. Similarly, the probability that the incidence rate is less than 12% is still quite large, with about 83% of that distribution lying to the left side of the 12% incidence rate. Another example. Here the key question is, are exposures in children similar to adults with this dosing rule? To dig into a little more specificity, and in a quantitative way, we could say, what's the probability that at least 90% of patients will be above the trough target with this dosing rule? That tells us some very specific details that we'll need to incorporate in the model building and simulation process, including some way of achieving a probability distribution, performance at describing percentage of individuals, and that a trough concentration prediction is important here to be compared to some reference point. The example shown below illustrates a couple of simulations, which again presents the results in terms of a probability distribution of the percent of patients above target. On the left-hand side, the probability distribution here is one that's centered around maybe 85%, but it doesn't have much of a density beyond the 90% target, which is part of our criteria. So there's a low probability that this dosing rule will achieve 90% of patients above the trough target. On the other hand, on the right-hand side, a slightly different rule for dosing pediatric population shows us that we have quite a bit of density in that probability distribution to the right of the 90% criteria. You can also apply this sort of quantitative specification across multiple endpoints, where the decision is informed by the probability of achieving multiple criteria. For example, this is one where we're attempting to select doses for a phase 3 trial of a new GnRH antagonist in endometriosis. The goal is to balance safety and efficacy with a high probability of meeting the target product profile. The key safety endpoint here is to avoid bone loss, so minimize the decrease in lumbar bone mineral density. The key efficacy endpoints are a reduction in non-menstrual pelvic pain and a reduction in dysmenorrhea. The endpoints then can be viewed each independently with different criteria, and we might specify quantitative criteria for some minimal threshold and maybe an optimal threshold, where in this case here, the first efficacy endpoint is viewed as a dysmenorrhea response rate at week 24 that's greater than 80%. A slightly better target might be to shoot for greater than 85% response rate. Similarly, we define quantitative criteria for the non-menstrual pelvic pain efficacy endpoint and the lumbar spine bone mineral density safety endpoint. These can then be incorporated in some sort of a summary metric, even potentially weighted differently depending upon the importance of these endpoints to the decision making. Those particular weights are hypothetical right now, and we'll get into a little more description on that sort of a strategy a bit later. So understanding the question and the specific quantitative criteria that are important in making the decision is really key. Katerina Fake tells us that so often people work hard at the wrong thing. Working on the right thing is probably more important than working hard. Or maybe in other words, your job is a lot simpler if you're focused and attentive to the elements that are most important in the decision-making process. Next, let's move on to identify knowledge sources and fill potential knowledge gaps. When we consider MIDD approaches in drug development, there are a variety of sources of prior knowledge. They might include internal proprietary data from preclinical or clinical trials, or maybe summary data that's been submitted for regulatory review, or if you happen to be working from inside a regulatory agency, maybe that's individual level data that's available for analysis as well. Other public sources include disease registries, extracting data from the medical literature or conference proceedings, maybe even gathering competitive intelligence from market communications and investor communications, or regulatory review documents that are published under Freedom of Information. Further, there are other sources such as real-world data, electronic medical record databases, data sharing consortia, or agreements that allow for a variety of data types to be accessed and applied to decision-making processes. And even expert opinions, which is a common practice in pharma development, to pull in a panel of key experts who rely on their medical experience to guide decision-making. These prior knowledge sources can come in a variety of shapes and sizes. They can be supplied as individual level data, sometimes summary level metadata from published studies, for example, mean and standard deviation of a particular endpoint. Sometimes a parameter is published based on some data analysis or model-based method, or even the entire model, mathematical structure, and key parameters are published. All of these sources are potentially relevant for an MIDD approach. This idea goes beyond the concept that a single trial is a source of truth for any decision. Instead, it attempts to integrate knowledge across a variety of information sources and acknowledges that a trial is just one data point in the context of a broader prior distribution of information. When considering the available knowledge sources and the type of insights that you hope to gain from the model-informed decision-making process, it might become evident that there are gaps in that knowledge. Maybe there are studies yet to be conducted in a particular patient population or at a particular dose level. Or maybe there's a key understanding of the disease state and the variability around the patient population in that disease state that is not well understood. Or maybe you have disparate pieces of knowledge across multiple clinical trial data sets or external metadata sets that have yet to be interpolated, integrated, and synthesized into a usable structure. Well, here's a flow chart that might help us to navigate through that process of identifying those gaps and what to do once we've identified them. The first question would be a relatively straightforward one for a modeling and simulation endeavor. Can the gaps be filled by a model-based analysis, given the current data and knowledge? Let's take the path to the right here, which is one where we'd answer no to that question. So the current data and knowledge don't allow us or are not sufficient to support a model-based gap-filling exercise. One of the options then is to define an assumption that links important knowledge sources together or that is key to bridging to the scenario that we hope to simulate or make a decision around. Another option might be to generate additional data. Maybe instead of applying the model-informed drug development strategy at this time, it's necessary to do another experiment or generate data from an available public database. Given those options, you might then move to develop models that are conditioned on the assumption or built from the new data that's been generated. On the other hand, if you had answered yes to that first question, the next question would be, well, do we already have models in place that fill those gaps or integrate knowledge sources? If the answer is no, we're back in the same position that we were just arrived at, and that is, well, there's a model development activity necessary to do that. If not, and the models do exist, then there is an additional step before you start applying the models, and that is to determine if the models are fit for the purpose intended. So whether the models exist or you're developing new models will end up at evaluating the suitability for the intended purpose. If that assessment identifies that the models are not well suited, then there's some more work to do refining those models in an iterative cycle until the models are suitable. Whether they're assumption-free, which is rarely the case, or dependent upon key assumptions, the models would then be applied as a simulation tool to explore different scenarios, different decision paths in an attempt to address the key questions. So let's go back to one of those earlier examples where we considered toxicity at a particular dose. What are some possible knowledge gaps here? Well, we didn't talk about the measures of toxicity. That was just some general summary. And maybe there are some toxicity measures that are more important than others, driving differentiation from competing therapeutics, or maybe more important to the patient population. The other question that's a gap here is the magnitude of the criterion. Is 30% something that's arbitrary, clinical intuition? Was it based on some prior data analysis of standard of care? And we never really mentioned what's the duration of treatment here that we're considering with respect to the tox incidence. Some other questions might be focused around the models. Is there a population pharmacokinetic model to support predicting exposures at this dose level? Is there a dose or an exposure response model relating dose to the probability of toxicity? Is it well suited for this purpose? These are all potential gaps that need to be evaluated. Here's another example, one in the context of establishing doses to bring into a phase 3 study. Linzagolix is a GnRH receptor antagonist being developed for endometriosis and uterine fibroid symptoms. This ties back to one of the quantitative criteria slides that we spoke about a few minutes ago. The symptoms of endometriosis are dysmenorrhea and non-menstrual pelvic pain. And the other problem with this class of drugs via this mechanism is the suppression of circulating estradiol, which might result in osteopenia. It's also the causal mechanism for the efficacy of this drug too. In this particular program, there are some key knowledge components that are well understood. The effects of efficacy and safety mediated through changes in estrogen. The relationship between estrogen and bone mineral density via previous off-the-shelf models, in this case a quantitative systems pharmacology model. And there's been prior work understanding the pharmacokinetic relationship for Linzagolix with a population PK model. On the other hand, we do have some gaps, and that's an understanding of the drug concentration to estrogen relationship, and then subsequently the estrogen to efficacy relationship. In further evaluating knowledge gaps for this particular example, we could confirm that some of the observed data is consistent with prior knowledge sources. In this case, the prior knowledge source is a quantitative systems pharmacology model that allows us to project a change in lumbar spine bone mineral density compared to the observed data under a range of doses for this GnRH antagonist. Here you see that the model-based projection in red is in good agreement with the observed data from this phase 2 study. But still, some gaps remain, and we do have data in this particular scenario from a phase 2 study that would allow us to construct new models relating the estrogen response over time to the dose of drug, as well as the pain reduction as a function of circulating estradiol. This is sort of a knowledge integration activity in that the available trial data were used to generate these quantitative relationships. So now that we've completed the exercise of identifying questions, key quantitative criteria, relevant prior knowledge sources, and the gaps in our current state of knowledge, and even integrated the knowledge or bridged gaps through quantitative models or key assumptions, we're ready to get to work. We're ready to apply the model now as a simulation tool to explore scenarios that are key to the questions that we're asking. Here's an example of extrapolation from adults to children. This case is centered around secukinumab, and it's a model simulation that's derived from a published clinical pharmacology review. This is a drug that's been shown to be safe and effective in adults for multiple psoriatic indications. And in our hypothetical drug development program, we're asked the question, which key doses are appropriate in a pediatric population? After further evaluation, we understand that we're projecting into a pediatric population without any data observations. And so we want to be careful that we understand the key features of that extrapolation. More specifically, we might say which combinations of doses and weight groups are necessary to achieve pediatric exposure variability that is consistent with the 90% population prediction interval from the adult exposures at the labeled dosing regimen. In other words, can we match the variability in exposure seen in adults by exploring a dose or multiple doses potentially administered in different age or weight categories for the pediatric population? This is a perfect setting for simulation-based scenario analysis. What we're going to do in the next few slides is explore the impact of dose and weight categories on the resulting expected exposures and response for secukinumab in a pediatric population. Let me orient you to these graphs. On the top left here, we have a simple plot that illustrates the dose level, 75 milligrams here, across the pediatric weight range. You'll notice it's the same dose level across all weights here. This is a one dose fits all type of strategy. Next we have, that's a simulation input. Next we have some simulation outputs. What we see in this panel is a relationship between trough concentration and grouping, weight grouping here. Now, in this example, we have only one weight group where all children are dosed with the same dose. It's just jittered here for appearance purposes. Each of these red dots represents an individual child trough concentration from a population of maybe 1,000 individuals that are simulated from a previously developed population pharmacokinetic pharmacodynamic model. So the red dots here indicate the scatter in trough concentration that we'd expect by administering a single dose of 75 milligrams to all children, no matter what their age or weight. The dark shaded gray region here is the range of variability that's associated with safe and effective doses in adults. The dashed line is the median trough concentration in the adult population. So we'd like to align the variability in the pediatric exposures somewhere within the adult 90% variability window. Next, you'll see a concentration time plot for a particular dosing regimen using a dose of 75 milligrams for all weight groups. This is a mean plot. And then further to the right, we see the pharmacodynamic response, a PASI 75 outcome over time under this particular regimen when dosed 75 milligrams to all pediatric subjects. So the question you might ask is, more than one dose group needed here, or is this sufficient? How do we know it's sufficient? Well, we haven't really set specific criteria other than we want to match the variability. Well, we see that there are some outliers above the adult target range, and there's certainly a good density below it here. The whiskers here indicate about 90% of the data. So this particular strategy is not too far off, although we do risk underdosing a fraction of the population, and a few individuals might be overdosed relative to the exposures established in the adult trials. Well, what if we were to consider additional groupings for the dosing rule? So now we have weight bins below 25 kilograms, 25 to 50 kilograms, and then 50 to 100 kilograms. And we've selected different dose levels for those groups, 25, 50, and 150 respectively. This is, again, a model-based simulation to explore the expected pediatric trough concentration variability under that dosing rule. And we see here that, although this does okay for the heavier patients, the lighter, smaller individuals are not well suited by this particular dose selection. In fact, you see that in the concentration time profiles, that the two lower weight groups achieve concentrations that are significantly lower than the higher weight group. And that's also manifested in the attainment of a suitable pharmacodynamic response, where we're aiming for somewhere around an 80% PASI 75 score. You could refine this further and look at another scenario here using the same weight group cuts, but now adjusting the dose for each of those groups to 50, 100, and 150 milligrams. This approach seems to do quite well and achieves both a concentration time profile that's similar across the different weight groups, as well as a pharmacodynamic profile over time. Let's continue with simulation-based scenario analyses. This example is an anti-effective where the key question is which loading and maintenance doses will provide adequate exposure coverage over the MIC. Quantitatively, we might translate that question as follows. Which loading and maintenance dose scenarios will achieve a free AUC to MIC ratio of at least 30 for at least 90% of individuals at both 24 hours and 72 hours after starting a once daily dosing regimen? That additional specificity in the quantitative translation identifies what we need to understand from our prior knowledge, where the gaps might be, and what sort of models we'll need to build to achieve this. Now imagine that we've done that, we've gone through the knowledge sources, gaps, and built models, and now we have a simulation tool to explore the scenarios. Let me orient you to this particular simulation visualizer. In the upper right panel here, we have an option to select the maintenance dose and the loading dose. These are administered, the maintenance dose once a day and a single loading dose at the beginning of therapy. Those are inputs to the simulation, and those are a couple of the variables that we can explore in evaluating a suitable dosing strategy. We've got a couple of other controls here, the fraction unbound, the MIC for the infective agent, and the pathogen, whether it's gram positive or gram negative. Now, these can be assumptions in the model. They can be pieces of information that we don't necessarily have a good handle on, but could be evaluated as assumed components of the model. The simulation is a pharmacokinetic simulation where that dose is converted to an exposure. In the first panel, we see the population distributions of the free drug concentration area under the curve for day 1 in purple and day 3 in green at the specified doses. That is then translated to a distribution in the population for the free area under the curve, or free AUC, to the minimum inhibitory concentration MIC ratio. The free AUC to MIC ratio is demonstrated for the population distribution on day 1 and on day 3 with a target of 30 identified by this dashed line. And then finally on the right panel, we see the percentage of individuals in the population that attain the target of 30 on day 1 or day 3, with our goal being this red line here at about 90%. So under this scenario, we can probably quickly come to the conclusion that a loading dose of 400 milligrams followed by 200 milligrams daily is not going to achieve our target. What if we try something else? This scenario now uses a loading dose of 400 milligrams and a maintenance dose of 400 milligrams. There's some improvement there, but we still see in the load that the day 1 fraction of the population achieving target is lower than we'd like. We can further explore that by thinking of maybe a larger loading dose, 600 milligrams on day 1 and 400 milligrams as the maintenance dose. And we see that our target is attained now both at day 1 and at day 3. Now this is a hypothetical simulation. Of course, when we do this sort of a simulation, we probably want some metric balancing safety or tolerability, but from an exposure-driven target attainment point of view, this is a simple way to explore the dose dependence. You'll notice that in this scenario evaluation, the assumptions have been held constant here. The fraction unbound, the MIC, and the pathogen type. It's necessary to do that when evaluating the particular inputs, although you'll see later that we can explore the sensitivity to some of those assumptions and how they might impact our decision about dose selection. By the way, the slides shown here with these simulation scenarios are actually generated from a web-based interactive simulation tool. This is a tool that we can construct by building a code-based model and simulation scenarios behind the scene that can be controlled through this web-based interface on a cloud computing platform. This is available for anybody in this course to explore in more detail at your own time via the links in the bottom of these slides. We often build these types of interactive simulation tools as a way to make the complexities of a model-informed decision-making process a little more accessible to decision makers by allowing them to explore different inputs and the resulting outcomes. There's this inherent sort of understanding of the system dynamics that results from these interactive experiences. Let's look at one more example of a simulation scenario analysis. This goes back to that prior concept of making a dose selection decision across multiple endpoint or criteria. In particular here, we're thinking about which doses should be selected for phase 3. We might translate that quantitatively in a simple way. For each dose, what's the probability of meeting each of the efficacy and safety endpoint criteria? What's the combined clinical utility at each dose when you summarize across all of those endpoints? Again, we've seen this sort of quantitative specification before. I'm just going to highlight that this is an opportunity to engage multiple stakeholders in the decision-making process, where the specific criterion for each endpoint can be defined in conjunction with the decision makers. In addition to the endpoint itself and the characteristics around that endpoint, the relative importance of each endpoint in the overall decision-making process can be defined by applying some endpoint-specific weight. Through modeling and simulation, we can derive a model-based probability of meeting each criterion and then summarize all of those in a weighted clinical utility function. I know that sounds a bit technical, but this is the same set of principles that we've been following all along. We're just combining these now across multiple endpoints. Let's take a closer look. This is actually a real-world example of a investigational therapeutic in rheumatoid arthritis, where multiple efficacy and safety endpoints were defined with both minimum target effect sizes and optimal target effect sizes, with also relative weighting applied to each one of these endpoints, so that when combined, the resulting utility over all the endpoints would reflect the relative importance of each of the endpoints. On the right-hand side here, we see model-based estimates of the probability of reaching the target effect, either the minimum target effect or the optimum target effect, for both efficacy and safety types of endpoints. These are then combined according to that weighting specification in a single summary function that could be used to select dose. Here you see the utility plotted versus dose. And we have two curves. One was considered for daily dosing in the red and twice daily dosing in blue. The sensitivity analysis we're going to explore in this example is sensitivity to decisions about how to weight each of the endpoints. This particular curve here reaches a maximum somewhere between 100 and 200 milligrams daily for both curves. And it's one that's derived when the efficacy and safety endpoints are all weighted equally. But maybe there are different points of view with respect to how to weight these endpoints in a utility function that's driving the decision. What if the only endpoints that were to be considered were the efficacy endpoints? Probably not likely, but it gives you a nice illustration of what you'd expect here. At the lower doses, the utility is low and it increases as doses increase, reaching some plateau. It's not going to get any better than that because you've reached 100% of the target. at least with the BID dosing, and over 90% with the QD dosing. But there's nothing in the utility function to counteract the dominance of the efficacy endpoints here, because the safety endpoints are excluded or set at a weight equal to 0. Similarly, we could explore a safety dominant view of the dose selection, where the lower doses, of course, are going to show the highest probability of meeting the target, but then as you achieve higher doses, that starts to drop off. Interestingly, though, that inflection point here, even with a safety dominant sort of weighting strategy, covers that range between 100 and 150 milligrams per day with a 90% utility. And then finally, if you asked different stakeholders on the team to weight the endpoints themselves according to their own subjective relative importance, here's the resulting curve where the utility rises with dose, reaches some sort of maximal level, and then begins to decline. The interesting thing about this exercise is that certainly you wouldn't make a decision purely on efficacy or purely on safety, but going through this sensitivity analysis results in a conclusion that doses in this range here between 100 and maybe 150 to 200 milligrams per day seem to achieve a maximum in the utility independent of the weighting scheme. And with that sort of a sensitivity analysis, we can be comfortable potentially in selecting a dose at 150 milligrams per day with the understanding that it meets the maximal utility under a variety of opinions around the relative importance of the different endpoints in this decision. Next, we'll move on to the topic of checking sensitivity of the simulation results to assumptions and uncertainties in the model. Dealing with uncertainty in decision making is something that we face whether we're using models or just general intuition. The challenge is doing something about it. Samuel Johnson's quote is particularly appropriate. Nothing will ever be attempted if all possible objections must first be overcome. The fact is, in drug development and in clinical therapeutics, decisions are made in the face of uncertainty on a regular basis. With respect to MIDD, we know that assumptions are fundamental to modeling and the application of those model-based approaches to drive decisions. We also know that the impact of the assumptions and uncertainties on model-informed decisions can vary. In other words, not all model-informed insights are equally reliable. Just imagine a projection of clinical efficacy that's made early in the translational preclinical research phase. Now, that might be based on biomarkers and competitor compounds that have made it further into the clinic, but that's a long shot, and the uncertainty associated with that projection is much greater than one that might be obtained after completing a phase 2A trial. Model-based projections should consider that. So the recommendation is to be transparent about assumptions and uncertainties. That goes a long way to gaining credibility in the use of modeling in general. Validate assumptions where possible against observed data or other sources of knowledge or information, if that exists. Account for model and parameter uncertainty in the simulations as they're being executed. And when communicating to decision makers, convey the impact of assumptions and uncertainties on the model-informed decisions and guidance. Sounds straightforward, and I'll share with you a few principles and examples on this topic. I'm going to ask you to recall a concept that was presented earlier in this training series, this idea of model-based projections and the parallel that we apply in drug development is also seen in weather forecasting. We've all seen this before. There's a tropical storm or a storm turning into a hurricane early on in its trajectory, And there's always a wide range of possible outcomes for where that storm might hit land. As the storm approaches, that cone of uncertainty is often reduced, and there's a better handle on where that storm is tracking. That's a similar scenario to drug development. Early on, when decisions are being made, the level of uncertainty can be much greater than after more knowledge is gained, collected, and integrated. Just like in the weather forecast, that particular uncertainty is acknowledged. It's still useful in understanding where the potential landfall might lie, but it's not until that uncertainty is reduced a bit that the model becomes more predictive of what's going to happen. So we're used to that acknowledgement in weather forecasting and other types of model-based projections. We can carry that sort of concept into drug development, because there certainly are multiple sources of uncertainty in MIDD approaches. One is simply the sampling-based uncertainty that's associated with observing data in a study, which is comprised of a sample of the entire population. That also leads to uncertainty in model parameter estimates that are derived from that data set. The models themselves make assumptions, and there may be some uncertainties in components of the model. For example, it's often necessary to make assumptions about the distribution structure of random variables in the model. Relationships between a predictor and a response, maybe exposure and response, can sometimes be quite empirical, but we might assume structures that are consistent with our understanding of pharmacology. For example, a typical Emax relationship. However, those structures do carry with them some uncertainty and should be evaluated given the observed data and all other sources of information. And in particular, if we're building mathematical models that are more mechanistic in structure, in some cases, our biological understanding is not complete, and the proposed mechanistic relationships may be an oversimplification or maybe even inaccurate. Nevertheless, those assumptions about those relationships may be necessary to proceed with some sort of model-based projection. Where possible, we try to validate these assumptions given data or other sources. If it's not possible to validate these, we're in the realm of exploring sensitivity of our conclusions to the assumptions and uncertainties. Here's an example of a model-based analysis with a simulation-based projection aimed at understanding the percent of patients who are misclassified with their systolic blood pressure using cuff blood pressure measurements at clinic visits versus an ambulatory 24-hour blood pressure monitoring. This model was informed with data from both cuff measured systolic blood pressure as well as ambulatory continuous measurements, and the associated variability with those measurements was calibrated, as well as the physiologic variability in blood pressure over the typical time of day. One thing that you see represented here in the model-based projection is an expected percent of patients to be misclassified at different times of day, and an associated probability interval around that. This represents the uncertainty in that projection. And for this particular example, the uncertainty is low, probably due to the fact that the model was informed by very rich data and there was good insight around this misclassification phenomenon. Here's another drug development example where the uncertainty and sensitivity of conclusions to those uncertainties was formally evaluated. This is a case of a fusion protein, which is the FC osteoprotegrin fusion protein for treatment of osteoporosis. Data are available from a phase 1 dose ranging study in healthy postmenopausal women. And in addition to pharmacokinetic data, a mechanistically relevant biomarker, urinary N-telopeptide, was measured, normalized by creatinine because of the urinary measure. That's known as NTX. With this information, the question was, which dose should be selected for the next study in patients? Probably a phase 2A type study. Quantitatively, we might say, for a given dose, what's the probability of achieving a mean urinary NTX response of at least 25% reduction at 2 weeks post-dose? Now again, the specific details here are derived based on some understanding of what the development team is trying to achieve, but the specificity is helpful in that it tells us that we need to understand the probability distribution for a mean response. There's a magnitude that we're targeting and a time point as well. These are plots of data from that study that includes single dose pharmacokinetics and pharmacodynamics. Here we see subcutaneous administration and the concentration time profiles at a variety of doses. There's also intravenous pharmacokinetic data, which is not presented here. On the right side, we see the urinary NTX versus time relationship for the individuals in this study across different doses. Again, you see a rapid drop in urinary NTX with some return to baseline evident in all dose groups over time. Given these data, a repeated measures nonlinear mixed effects model for the pharmacokinetics and pharmacodynamics was developed and evaluated for fit for purpose use to simulate new scenarios. Here's a resulting simulation. For multiple dose levels, a similar simulation was conducted to explore the expected change from baseline in urinary NTX over time. We're presenting one dose level here. And what we're observing is, there are multiple lines in this plot. What we're observing is a mean response indicated by the dark solid line, plus some uncertainty interval around that, some measure of the precision of that mean, as well as the population variability, which is denoted by the lighter solid lines, and some uncertainty around that as well. This is an informative simulation in that now we can understand quantities describing the central tendency, the median response in the population variability described by the expected 95% prediction intervals in the population, and also the associated uncertainty or imprecision in those projections. Again, like we saw with the blood pressure assessment model, the precision around these projections is relatively narrow, indicating that there's good information to support the projection of this model for this purpose. A nice side effect of conducting this sort of a simulation is that the results are viewed in terms of a probability distribution. That probability distribution can be extracted at any point in time that's of interest. So if we take the 2-week time point and we take a slice of the median response here and look at that distribution. That's what we have on the bottom left panel. This is the probability distribution of the change from baseline in NTX at 2 weeks for the median individual. That can be compared versus some reference point here. We defined up front that it's a 25% drop. And if you look at that line here, you'll notice that the majority of this distribution at 2 weeks is less than a 25% change from baseline. So we've achieved the target goal at this dose of being at least 25% decrease in NTX from baseline. That could be attached, there could be a probabilistic statement attached to that, and it would be a high probability here, maybe 95, 99% probability that this dose results in the desired response at 2 weeks. Now that's just one consideration in dose selection. Of course, there would also be potential tolerability and safety endpoints to consider. But let's continue forward thinking about the conclusion based on an efficacy point of view. What if we didn't really understand the parameters of the model with very high precision? What we're showing in these plots is the change in NTX that would be simulated versus two key parameters. One is clearance. This is the primary pharmacokinetic parameter. And one is IC50. It's a key pharmacodynamic parameter describing the sensitivity, or in other words, the concentration at which half maximal inhibition would occur. Clearance and IC50 are parameter estimates, and these are the population average values. But in this analysis, there's some uncertainty in the estimate of population clearance. And this reflects maybe a 95% interval around the clearance. It might range from 150 to 180 mils per minute. IC50 is similarly estimated with some imprecision, and again, the range of IC50 values shown here reflects some measure of precision around the mean IC50 estimate, ranging somewhere from 2-ish up to 8 or so, whatever the units are. By incorporating that uncertainty or the lack of knowledge about these parameters in the simulation, you necessarily generate an understanding of the sensitivity to the endpoint we care about. This is mean change in NTX at a particular time point. And we can visualize how influential these parameters are and to what degree our uncertainties in these parameters might lead our decisions astray. On the clearance side, you see a relationship that reveals very little dependence. In other words, the change in NTX is pretty much insensitive to whatever the population clearance would be at the doses being studied here. Whereas on the right-hand side, there's a strong relationship between the change in NTX and the population IC50. So if our point estimate is 5, we end up somewhere around a drop of let's say 60%. But if we're wrong with that, and point estimate's closer to 3, well, we can increase the response to maybe a 70% drop. Or if we're at the other end of the extreme here, maybe 7 or so, we're only achieving about a 55% drop. So this is an objective assessment of the sensitivity of the endpoint to uncertainties in these parameters. But how does that impact our decisions? Does it really matter? Well, that all depends. It depends on what kind of decision criteria we're applying to the question. If the question was, which dose gives us at least a 50% drop in urinary NTX at this time point, so the horizontal green line, we see that in both relationships here, there's a high probability of no matter what the value of clearance or IC50, that we've achieved that objective. So the sensitivity that we see on the right-hand side is really irrelevant because the decision is not influenced by the insensitivity at that particular metric. As you can imagine, if we changed our target and now said, okay, what about the probability of achieving a 60% drop? Well, on the left-hand side, we see that that probability is still quite consistent. You have about half of the simulations exceeding that response, and it's independent of the value of clearance. On the other hand, with IC50, if we're wrong with IC50, and maybe the true value is much higher, we'd say there's a low probability of achieving that target. At the midpoint, there's about a 50-50 chance. And at the low end of IC50, there's a high probability of achieving that target. So what do? We basically end up with different conclusions depending upon our understanding of the IC50 parameter. One thing that could be done is to improve our understanding of IC50. In other words, narrow this range of parameter uncertainty to some tighter interval that is more consistent with respect to the NTX change from baseline. These sorts of decisions in drug development happen all the time, in that some conclusion is necessary to be made even when there's some uncertainty. So it might not be feasible to wait and conduct another study where we can more accurately refine the estimate of IC50, and there's maybe an opportunity to move forward at a higher level of risk, but at least that risk is understood given the sensitivity we've just demonstrated. We can look at sensitivity in other ways too, just based on fixed values of assumptions in the model. Remember the anti-infective target attainment simulation. We ended up at a loading dose of 600 milligrams followed by 400 milligrams daily to achieve 90% of patients at target. That particular simulation was based on some assumptions defining the fraction unbound in plasma of this particular drug, the MIC of the pathogen with this agent, and also the pathogen type. Imagine that we're early in development and we haven't fully characterized the ADME properties of this compound, and so maybe the free fraction is not well known. We can explore that sensitivity to that value by changing the assumption. Here we've moved it up to 80% fraction unbound, and you see at the doses that we've already concluded from the simulation scenario analysis that we still meet the criteria if we change the assumption. However, if we change that assumption to a lower fraction unbound, we see that our conclusion about this dose regimen reaching our target is no longer valid. So we've demonstrated the sensitivity to the assumption about fraction unbound. The next step would probably be conducting an experiment or seeking other sources of information to refine our estimate of fraction unbound, given that it's a highly influential assumption in our simulation-based assessment of the dose selection for this anti-infective. To conclude, I'm going to run through a few examples of MIDD in practice, highlighting some of the key points in the MIDD strategy that we've been talking about in the prior module and in this one. Let's go back to the linzagolix example. This is the GnRH antagonist in endometriosis. Remember, we've talked about how to specify the question quantitatively. We looked at knowledge sources and how to assess knowledge gaps. And now we're at the point of applying the model for a simulation-based decision guidance. The models were applied then to explore the relationship for the safety endpoint, lumbar spine bone mineral density, to Lindsay-Gulick's dose with a typical median response and some probability distribution around that. Again, reflecting the uncertainty in that projection. The same sort of concentration response relationship is presented for the efficacy endpoints. This time it's the probability of pain reduction versus estradiol concentration, both for the non-menstrual pelvic pain and dysmenorrhea pain. These relationships characterize a higher probability of pain reduction as the estrogen concentrations are lower. This is a known pharmacologic expectation, but what this also does is quantify the typical relationship and the uncertainty around that, so that we can propagate the relationship as well as any uncertainties around that into the simulations. So a decision around dose selection can be recommended given these models and the associated uncertainties in the model-based projections by defining clinically meaningful targets for the endpoints, BMD and pain reduction. Table 2 here illustrates the target for the percent of responders with respect to the pain endpoints and the mean change from baseline in lumbar spine bone mineral density that is tolerable in this therapeutic. On the right-hand side, you see now probability curves as a function of dose for each of the endpoints. These are made possible by that probabilistic projection we saw on the prior slide. And you see, as you might expect, the pain responses in red and yellow start at some lower value at lower doses and increase to some plateau with respect to the probability of meeting the target number of responders, whereas the safety endpoint has the reverse opposite shape, being maximally probable of achieving that safety target with a decrease in that probability as doses increase. In the middle, there's a sweet spot, and potentially a dose around 100 milligrams maximizes the probability of achieving both of the efficacy endpoints and the safety endpoint. We can apply a model-informed decision-making process to the development of trial designs as well. Here's an example of a population PK study design for actinomycin D in a pediatric population. The goal here is to learn from existing data and design a study that will inform the understanding of the population pharmacokinetics of actinomycin D in a younger patient population than had been previously studied. The prior knowledge includes pharmacokinetic data across a range of body sizes in prior studies from children and young adults, and also an understanding of the model-based description of those data. The model in this case is a traditional intravenous input with a multi-compartment disposition. 3 compartments were necessary to describe the kinetics of actinomycin D. The simulation-based evaluation of the trial design was initiated by exploring potentially informative designs, and also balancing that with what would be clinically practical to execute. That resulted in a nominal design characterized by a total population of 100 children, randomized to two groups of sampling in order to provide some variability around the sparse PK sampling profiles. The first group had a very early sample between 5 to 15 minutes post-dose, then followed on between 3/4 of an hour to an hour and a half, 3.5 to 4.5 hours, and then for a subset of individuals, a late time point out beyond 48 hours. Group 2 is similar in that there were 4 sparse sampling times, but those times were slightly different from the Group 1, allowing a more complete characterization of that time course across the population. So the first time point's a little bit later, as is the second and third, with a similar terminal time point somewhere in that 48 to 96 hours post-dose. The quantitative question that we're exploring through simulation is about the design. So which design results in adequate characterization of PK? More specifically, we're going to ask for estimation of mean population PK parameters, clearance and central volume of distribution, that are within plus or minus 10% bias from the true value. Now, the nice thing about simulation is that we can always anchor on some measure of the truth. In this case, the true value is the value used as the reference point for the simulation. What we'll do is then apply an estimation-based method to try to extract the true parameters of the model. Here's the result of that exercise. What's plotted is the percent bias in the estimate of population typical clearance and bias in the estimate of the population mean volume of distribution. This is illustrated, like we saw earlier, as a function of the imprecision or the uncertainty in the population clearance and volume. We can conclude with respect to the performance of this design for clearance that there's a high probability, and we can actually be quantitative about the number of data points here, lying between plus and minus 10% bias. There's some sensitivity to the clearance estimate, but not overly sensitive. On the other hand, the bias in the estimate of central volume distribution is extremely high. In fact, you see our reference point of plus 10 is shown here. We're not even including the minus 10 because across the entire range of potential values for volume, this trial design performs poorly or outside of our target range. So again, even though there is some sensitivity where if our population volume is low, maybe 2 liters versus a higher number, the entire range of results is outside of our decision criterion. So we can conclude with confidence that despite there's uncertainty in estimation of clearance and volume, this design is not adequate for estimating our volume distribution with the desired metrics. So what do we do? Consider an alternative design. And given that the central volume of distribution is highly informed by those early time points, you'll see that this design actually adds an additional sample and focuses on some earlier time points. In Group 1, we've got the 5 and 10 minute samples. In Group 2, we've got a 5-minute sample. And then spacing samples over time all the way out to potentially 96 hours. We've also increased the sample size in this simulation to achieve some additional precision around the estimates of those parameters. The thought process around this can be one driven by intuition, or there are also formal mathematical methods called information theoretic approaches that allow us to optimize the sampling time points. Again, the design has to be one that is practical from a clinical operations point of view. Let's see how this design performed. Now we see little dependence on the conclusion about bias in the estimate as a function of the uncertainty in our population clearance or the uncertainty in our population volume. There's a high probability of achieving the desired accuracy of the clearance estimate under this design, and there's also a high probability of achieving that with respect to the volume. Again, we could get quantitative about this and quantify the number of simulation results that lie within our region of interest versus the total. But you can see graphically that this is a drastic improvement over the prior design. Finally, we'll shift gears and talk about a different kind of decision. This is a proof of concept decision, that sort of inference that's made in drug development where sufficient confidence is gained that the new therapeutic will result in a meaningful clinical response in a patient population, such that further investment in the development program is warranted. The indication here is Alzheimer's disease. The therapeutic is a symptomatic treatment, and the study design is constrained by potentially budget or patient availability, such that the proof of concept study will be limited to 12 weeks. On the other hand, the target product profile, which was identified by the development team, is based on a longer term endpoint that specifically is 6 months. So there's a disconnect here between the empirical data, what we're able to collect in the proof of concept study out to 12 weeks, versus a target product profile or decision criterion at 6 months. The key question then is, will the treatment result in adequate efficacy? We can translate that quantitatively by saying, what's the probability that the median 6-month change from baseline in the ADAS-Cog endpoint will be at least a decrease of 2.5 units and optimally a decrease of 4 units. So we see that sort of probabilistic quantitative translation with a time point, definition of a median response, the specific endpoint, and the magnitude of change that's desired. We have 2 goalposts here, one that's the minimal requirement and one that would really be nice to have. One of the knowledge gaps that we acknowledged up front is that our observed data in this trial will not be of sufficient duration to assess the 6-month decision criterion. So we might look to other knowledge sources to augment the observed data and improve the model-informed decision-making at a 6-month endpoint. For this example, a disease progression model was developed from a variety of sources. This disease progression model allowed for the longitudinal characterization of disease progression, which was informed by natural history data at the individual level from the ADNI database, by the placebo response, which was informed by a shared database supported by the CPAD consortium. This included multiple trials and individual patient-level data for the placebo arms in these pharma-sponsored studies. And then the literature metadata, which described the typical responses for competing therapeutics that are symptomatic treatments of Alzheimer's disease. But given that these are from the published medical literature, we're limited to metadata, in other words, mean and standard deviation, collected longitudinally over time as published for these studies. All of these data sources were integrated in a longitudinal model that described the natural history progression, placebo response, and expected competitor response. Now we've got a framework to analyze the 12-week POC data. But instead of making a conclusion or a decision based on the observation at the end of 12 weeks, this model-informed decision-making actually takes the 12-week data, augmented with the longitudinal disease progression model, and makes a model-based projection out to the endpoint of interest at 6 months. The panel on the left shows the model-based projection for the effect on ADAS-Cog over time, up to the 12 weeks of the POC duration, and beyond, all the way out to 6 months. The empirical observation would be limited to this first 12-week period, but the model-based extrapolation allows us to make some information-guided decision about a 6-month endpoint. That projection is shown with a typical expected response and the associated uncertainty or probability distribution. You'll note that in this case, that uncertainty is rather large relative to some of the others that we've seen. And that's fair because we're making an extrapolation from 12 weeks out to 6 months, where that's not been studied yet for this new therapeutic. And we're also pulling in data from a variety of sources which all bring their own variability and uncertainty. When we view that probability distribution for the estimated effect at 6 months, we can then compare the projection to the decision criteria. Remember, we identified that we must have at least a drop in 2.5 units. That's this dashed line. And there's a pretty good density, or pretty good probability that the ADAS-Cog response will increase the drop beyond 2.5 units. That estimate is somewhere around 90%. When we think about the optimal value here, or a drop of 4 units, the density beyond that, or the probability of exceeding that, is smaller, but still relatively large at about 27.5%. This kind of guidance can then be used to select a dose, or even to consider moving forward with a trial as a potentially positive outcome. In addition to dose and probability of success for the trial, the design of the POC study could also be evaluated. Using the same model as a simulation tool, 2 different designs were considered here. The first is a parallel group design with patients randomized to placebo or drug treatment. The second is a crossover design, which randomizes patients initially to placebo or active treatment, but then after a washout, a crossover to the alternative treatment arm. Both studies are 12-week designs. Those designs were evaluated based on performance for making the right decision. The right decision in this scenario is that a drug that doesn't achieve the minimal criteria, so for example, the expected value at 6 months is only 2, we should decide that this hasn't met the target and we would not go forward with development. On the other hand, a drug that is clearly a winner or meets the criteria of a change of 4.5 units at 6 months, that's one where we'd want to move forward and develop. The grid represents the outcomes of the simulation study under the 2 designs, a 12-week parallel design versus a 6-week crossover design. You see the performance of these 2 is similar. In fact, it's the same when it comes to a drug that is truly effective. We've got about an 8% false negative conclusion here. 92% of the time, in both designs, we would elect to move forward for a drug that does have the sufficient efficacy to warrant further development. The place where these designs are differentiated is in weeding out those therapeutics that are not likely to have sufficient efficacy. So a response of 2 units at 6 months. The 12-week parallel design does very well here in that 100% of the simulations indicate that the program should not move forward. Whereas the 6-week crossover design has about a 10% false positive rate, indicating that 10% of the time, this design would result in a decision to move forward with development when really the drug does not meet criteria. It's interesting to try to understand why these designs perform differently. One intuition is that it's the time course on active treatment and placebo that's really necessary here. And so having observed treatment out to 12 weeks is more informative than the 6 weeks observed in the crossover study, even though there's a reduction in the comparison variability when individuals are crossed over. It's because I believe the decision process is based on a longitudinal extrapolation. Nevertheless, this illustrates how that theoretical understanding of the disease progression, placebo response, and potential drug effect over time can be used to inform dose selection, probability of success of a proof of concept trial, and the design of that proof of concept trial. So in the past 2 training modules, I've shared several principles about the application of MIDD, starting with the value of model-based approaches versus purely empirical data observation. We took that model-based theoretical understanding of the system and applied it as a decision-making tool through simulation. You've seen several examples and many characteristics that make model-informed drug development strategies effective. To summarize, MIDD organizes around a series of questions. Getting the questions right is key to deriving any useful model-based guidance. MIDD also aims to advance the theoretical understanding of the problem. As we saw in multiple examples, the model-based theoretical understanding can be used to drive new study designs, generate new data, improve the theory, and guide future decisions and explore unstudied conditions. MIDD quantitatively integrates knowledge and bridges knowledge gaps. It's that extension of the theoretical understanding to a bridge between a variety of knowledge sources, unifying them and integrating them in a consistent framework. MIDD allows for the exploration of alternative options. Contrast that with the point and interval-based estimate decisions that we spoke about earlier. True model-based approaches allow us to evaluate options and scenarios that were not part of the original data observations. MIDD is transparent about underlying assumptions. Oftentimes, decisions are made based on intuition, but that sort of cognitive intuition is subject to potential biases that are not transparent to others in the decision-making process. A mathematical model requires us to be very specific and transparent about the assumptions being made to evaluate the system and drive decision-making. MIDD assesses sensitivity of conclusions to the assumptions and uncertainties involved in the approach. Again, the transparency of those assumptions and uncertainties allows us to do that because we can then view the resulting conclusions and their sensitivity to the underlying assumptions and model-based uncertainties. MIDD delivers insights, and it does so with the associated probabilities of desired outcomes. This is a particularly useful way to deliver guidance to decision makers in that the probability of achieving some outcome is defined by the outcome that they've been part of defining from the beginning. I'm hopeful that you've found the principles and examples that we've discussed in the last 2 training modules to be informative and helpful in your understanding of model-informed drug development, both from a theoretical understanding of the system and an application of model-based methods to inform decisions. Some additional references are included for your further study, and I'll close by offering to answer follow-up questions directly, please just feel free to reach out to me. Thank you all for your attention, and I wish you continued success in your learning about MIDD.